Hello everyone, I'm Mr. Furlong, and today we're going to take a look at how DNA can make a copy of itself. Today we're going to take a look at how DNA can make a copy of itself. This is what is known as DNA replication. Of course, anytime a cell divides in the process of mitosis, the DNA has to copy itself so that each new cell gets a complete set of that DNA. Today's video is simply looking at how this process is going to take place. So let's get started. I'm going to start off by making a diagram of DNA and this process of replication. You can go ahead and make the same diagram in your notes. We will also be writing down some of the steps that's taking place. What you're looking at here is a drawing of DNA. The little triangle is representing an enzyme. In this case, the enzyme is called the DNA helicase enzyme. Now, the job of this little enzyme is to separate the two sides of the DNA molecule. Oftentimes, we say that it is unzipping the DNA. So let's draw what this looks like. Let's put some letters here. And recall, if we know one side of the DNA molecule, we know what the other side is because A's pair with T's and C's pair with G's. We can see as this DNA helicase enzyme is moving through the DNA molecule, it is separating the two sides of the DNA by breaking the hydrogen bonds between the bases. And then new nucleotides are going to be added on to both sides of the DNA molecule. Now, these new nucleotides are primarily coming from foods that we've eaten. Let's draw some of these nucleotides. Remember, A's pair with T's and C's with G's. Now, in order for these new nucleotides to be bonded together, another enzyme comes along and forms the bonds between all the A's and the T's and the C's and the G's, in which case then the DNA gets zipped back up again. So let's draw this. We end up with then two identical chromosomes. But the thing is, this happens really fast. And sometimes mistakes are made. So proofreading enzymes come along and they check to make sure that there really are the A's and T's paired up and the C's and the G's are paired up, that there isn't a base that maybe doesn't have a partner that's attached to it, and if it finds it, it will fix those mistakes. Now, this is a real simple drawing that I made here. Let's take a look at a video that shows this process in action.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
No, what happened to my voice there for a minute? Anyways, we mentioned before that there can be mistakes when the DNA is copying itself. Anytime there's an error in the DNA, that is known as a mutation. Mutations play a big part in evolution. We'll talk more about that in another video. But let me just show you how often mutations take place. You don't need to put this in your notes. This is just a little bit of information to get you thinking about how often this happens. Remember that in DNA, if we were to look at in a human all 46 chromosomes, there'd be about 6 billion nucleotides. And for every 1,000 nucleotides, there's one mistake. Now when you think about that, that's actually not very many mistakes. Imagine if we had a test with 1,000 questions on it and you only got one wrong. You would think that would be great. I know I would. But if there's 6 billion nucleotides, that means that there's actually 6 million mutations that take place every single time we make a copy of our DNA. How often? Remember, we make about 10,000 new cells every second. We're constantly making new DNA molecules or chromosomes. But remember, we have these proofreading enzymes, and they fix 99.9% of the errors. So again, imagine if you had 6 million mistakes. That doesn't sound very good, but you can go back and you can make corrections and you fix 99.9% of those mistakes. That's pretty good. Except for the fact that 0.1% of 6 million is 6,000 errors still left uncorrected. In other words, there's 6,000 mutations every single time we make a copy of our DNA. This sounds like a lot, and it is, but most of these cells that we are making, if there's a mutation, chances are it's not even in a gene. If it is in a gene, it's probably in a gene that we're not even using. But even if that mutation is in a gene that we are using, we have hundreds of thousands of other cells that don't have a mutation, and so we don't really see the effects of this. It's only when these mutations take place in genes when we are producing new sperm cells and egg cells, and by chance those are the cells that end up in the fertilization process. We then pass that mutation on to our offspring. So I hope you enjoyed that. In the next video we're going to be taking a look at how proteins are made, and I'll see you then.